Hello everyone. Today, in the subject of medical microbiology, we are going to learn about clostridium. So, what is the objective of this topic? Our objective is to give you a brief introduction about the clostridium and what are its fundamental applications and that is required to uh, in order to prevent and uh, do betterment for the humanity. So, Clostridium is a genus of gram-positive bacteria. Right. This genus includes several significant human pathogens, including the causative agent of botulism and tetanus. The genus formerly included an important cause of diarrhea, Clostridiodis difficalis, which was reclassified into uh, Clostridiodis genus in 2016. They are obligate anaerobes that are capable of producing endospores. So, Clostridium is very poisonous in nature, right? And it is very pathogenic in nature, and it has many variants that produce several types of toxins that are very much harmful for the human beings because they readily affect the physiology and they are very harmful in nature in the long term. The normal reproducing cells of uh, Clostridium, called the vegetative form, are rod shaped, which gives them their name from Greek uh, that means spindle, Clostridium. Clostridium endospores have a distinct bowling spin or bottle shape, distinguishing them from other bacterial endospores, which are usually ovoid in shape. So here you can see that uh, when you see the spores of Clostridium, they are in the shape of bottle. Okay, and normally the bacteria, the Clostridium bacteria, is in spindle form. So endospores they form almost very distinct structure. You can readily identify them from their shape. Clostridium species inhabited inhabit soils and the intestinal tract of animals, including humans. So their habitat is very much indifferent. They are not only found in soil but also in the intestinal tract of animals. Clostridium is a normal inhabitant of the healthy lower reproductive tract of females also. So it is not only found in soils and animals, but it is also found in healthy reproductive tract of females. So here in this diagram, you can see that Clostridia is an obligate anaerobe. Okay, that means oxygen is toxic for it, and it has to survive in absence of oxygen. So it is mainly found in environments where oxygen is not very much present. So what it does, the Clostridium forms spores. Okay, and you know that spores formation takes place when bacteria face an extremely harsh environment that is not favorable for its growth and that is not extremely supportive for its structure. So what does it do? It mm, begins the process of spore formation and everything that is important for bacterial formation um, just wraps itself inside the spore and spores are very tough. Okay, and for a very long duration of time they can survive in external environment until unless the environment does not become supportive to the bacteria. So as soon as the environment becomes supportive to the bacteria, uh, it immediately breaks the spores and gets released in the environment. So the spores are metabolically inert, resilient to the environment. Okay, they are also very tough to autoclave also. So further, they are gram-positive spindle-shaped bacillus, mostly present in cylinder or rod shape. And when they are forming spores, their spores are of um, bottle shape. They can readily be observed under the microscope. So clostridium botulinum produces botulinum toxin that causes botulism. So this image readily explains that how that functionality takes place in Clostridia. So here we have another image of Clostridium uh, perfringens. Okay, now what is the speciality of this Clostridium? It thrives in soil. Okay, so the family of Clostridia. Clostridium belongs to the family of Clostridia. It is obligate anaerobe. Okay, it readily requires to be uh, thriving in oxygen-less environment. Don't require oxygen to thrive. Lack the enzyme catalase or superoxidase dismutase. So the enzyme catalase and superoxidase dismutase is absent there. Oxygen is actually toxic to them. It is readily explained by obligate anaerobe. Okay, being an anaerobe is an obligation for them for their survival. So if they are coming into contact with oxygen, it would prove to be really very lethal for them. Produces spores that are resilient to the environment, can even survive cooking when food is slowly cooled or stored. Spores they form full-fledged clostridia. So one really has to be extremely careful when dealing with this type of clostridium because on the other hand, not only uh, it is another form of clostridium, but it is also really really very tough in nature. Okay, and it can not only survive the environment, but it is extremely resilient also. When food is slowly cooled and or stored, the spores they turn into full-fledged clostridia. So most of the time, the accidental cases of food poisoning or eating of toxic food materials are due to this only, because the food material that has been contaminated with Clostridium perfringens is just containing spores. Although slow cooking and storage leads to the full-fledged development of Clostridia in it, and whosoever is going to eat it, it would cause them extreme level of disease and food poisoning to them. The genus, as traditionally defined, contains many organisms not closely related to its type species. The issue was originally illustrated in detail by rRNA phylogeny from Collins in 1994, which split the traditional genus now corresponding to a large slice of Clostridia in 20 clusters. with clusters containing the type of species and close relatives. Over the years, this has resulted in many new genera being split out, with the ultimate goal of constraining Clostridium to cluster. So there are several different types of Clostridium. As the research is progressing, as the technology is progressing day by day, scientists have been able to differentiate them properly, right? So every time they just identify a new characteristic and they separates the bacteria from that very group. So it is extremely important that every time several different groups of bacteria are separated out, because they might be having some characteristics that are not very much essentially native to that very particular group, because bacteria they do look very similar but they are very different from each other, right? So every time what we need is that we have to differentiate between the bacteria uh, by following the procedure of complete study every time. So clostridium cluster uh, and clostridium cluster 4 and 14 efficiently ferment plant polysaccharides composing of dietary fibers, making them important and abundant taxa in the rumen and large intestine. As mentioned before, these clusters are not a part of current Clostridium, and use of these terms should be avoided due to ambiguous or inconsistent usage. So, Clostridium also have a variety of variants. Okay, that perform very various different types of functions, and they are not only linked with other things. Like here we have seen that they are efficient in fermenting polysaccharides, so that contains especially the dietary fibers, and not all of them are extremely poisonous, but many of them are also useful. So in this image, you can see the Clostridium botulinum and its spores that are being formed by it. Pathogenesis and classification. Clostridium contains around 250 species that include common free-living bacteria as well as important pathogens. So, Clostridium genre or the family is extremely large, and it has around 250 species that include common free-living bacteria as well as some other important pathogens. So you cannot say that only one single Clostridium is responsible for all the diseases. Okay, it's a very huge family, and they always have many significant and different functions. Okay, the main species responsible for human diseases are, so there are particular type of species that cause diseases in humans. Not all the species of Clostridium are pathogenic to humans. There are specific type of species that are extremely pathogen. Now we are going to study about them. Uh, Clostridium botulinum can produce botulinum toxin in foods or wounds and can cause botulism. Okay, it is a condition that is linked with central nervous system, and it is extremely dangerous. This is the same name. This same toxin is known as Botox and is used in cosmetic surgery to paralyze facial muscles to reduce the signs of aging. It also has numerous other therapeutic use. So, what scientists have learned, they have learned to utilize this toxin for the betterment of human body, and they have started using the botulism toxin to enhance the cosmetic surgery in order to paralyze the facial muscles to reduce the signs of aging that is commonly seen. So the individuals who are going through the cosmetic surgery, they do got they have to get the Botox uh, after the time period of 3 to 4 months. So it has numerous other uh, therapeutic uses also. So you can see that this large family of bacteria, this large family of Clostridium that has around 250 species that include many of the different types of bacteria that have several functionalities, and in long term they do help humans. Okay, there are many other uses. So pathotype, species, diseases. So a pathotype neurotoxic. Okay, Clostridium botulinum, and diseases it causes botulism. Clostridium tetani causes tetanus. Histotoxic, Clostridium perfringens. Okay, gas gangrene. Clostridium septicum, malignant edema, brachy. Clostridium chovy, blackleg. Clostridium novi, infectious necrotic hepatitis. And Clostridium sordeli, uh, and arthritis. So enteric, Clostridium perfringens, arthritis, enterotoxemia. Clostridium difficile, diarrhea, antibiotic associated diarrhea, and pseudomembrane colitis. So there are several other diseases that are caused by many other types of Clostridium. So when we go into the classification of this uh, family, what we see that they have a variety of nature. Okay, not the two type of Clostridium are similar. Every time they have differences, and their functionality is very different. So what we see there, classification, tetanus caused by Clostridium tetani, acute colitis by Clostridium uh, difficile, gas gangrene established by pathogen Clostridium perfringens, Clostridium septicum, etc., and less pathogenic Clostridium histolyticum and Clostridium falix, and doubtful path of uh, Clostridium bifermentans and Clostridium sporogens. And especially the food poisoning is caused by Clostridium perfringens type A and necrotizing enteritis by Clostridium type C, perfringens type C, and botulism by Clostridium botulinum. So, Clostridium perfringens causes a wide range of symptoms from food poisoning to cellulitis, uh, fasciitis, necrotic enteritis, and gas gangrene. Clostridium tetani causes tetanus, and Clostridium sordeli now can cause a fatal infection in exceptionally rare case after medical abortion. So, bacillus and Clostridium are often described as gram variable because they show an increasing number of gram negative cells as the culture ages. So, gram variable. So, these bacteria they are extremely dangerous in nature because your gram staining does not work on them significantly. Okay, so if we see the gram staining procedure, there are gram positive and gram negative. and in between them there are gram neutral or gram variable species. So most of the time they prove to be extremely lethal because you cannot significantly identify them, and they do not react towards the gram staining. So some clostridium species are also gram neutral or gram variable in nature. So it is defined as an exceptional condition, right? So uh, it should be dealt with extreme caution, and it should be observed carefully because in the laboratory conditions they should be treated with extreme caution because they show an increasing number of gram negative cells as the culture ages. So from being gram positive, their nature changes to being gram negative as the aging of the culture takes place. And if you're going to use gram staining on them, then they will show the characteristics of gram neutral in nature. Clostridium and Bacillus are both in the phylum Firmicutes, but they are in different classes, orders, and families. Microbiologists distinguish uh, Clostridium from Bacillus by following features. So Clostridium and Bacillus, they might do look very similar in nature, but there are some differentiating characteristics that makes them apart from each other. Okay, so Clostridium grows in anaerobic condition, right? So oxygen is extremely lethal for it. Bacillus grows in aerobic condition. For the growth of Bacillus bacteria, oxygen is extremely essential. So, for you can say that in other words, the Bacillus oxygen is important. For Clostridium, oxygen is really very lethal. So here you can readily observe the two cases. Uh, one where oxygen is extremely essential, and second where oxygen is extremely poisonous. So they readily say that Clostridium grows in anaerobic condition, so which is obligate anaerobic bacteria, and Bacillus grows in aerobic conditions, which is obligate aerobe. So these are two major differences between Clostridium and Bacillus. Clostridium forms bottle-shaped endospores. Bacillus forms oblong endospores. So when it comes to the study of these spores and the condition that leads to spore formation, Clostridium forms bottle-shaped spores. Okay, that is why you can easily identify the Clostridium endospores from other endospores. But the Bacillus forms the endospores that are oblong and oval in shape. So this is another factor that makes them different from each other. Clostridium, uh, Clostridium does not form the enzyme catalase. Okay, Bacillus secretes catalase to destroy a toxic byproduct of oxygen metabolism. So, catalase enzyme is a specialty of bacteria that lives in aerobic condition, and they readily produce it in order to destroy the byproducts that is caused by oxygen metabolism. But on the other hand, Clostridium does not have any role for oxygen, and oxygen is extremely lethal or poisonous for it. So it does not produce any part of catalase enzyme because it lives in the environment uh, that is free from oxygen, or oxygen does not play any role in its survival. So being it never has to deal with the byproduct of oxygen also, and it readily lives in an environment that is uh, totally absent from oxygen. Okay, but on the other hand, because the bacillus is uh, is more used to or obligate aerobic bacteria, so it requires a lot of oxygen for its metabolism and growth. So in the long term, it has to deal with the oxygen by metabolism byproducts also. So catalase enzyme is produced to destroy them. Clostridium and desulforta malacum are both in the class Clostridia and order uh, Clostridiales, and they both produce bottle-shaped endospore, but they are in different families. So Clostridium can be distinguished from Desulfoto malacum on the basis of the nutrients of the genus uses. The latter requires sulfur for its survival. Glycolysis and fermentation of pyruvic acid by Clostridia yield the end product, uh, butyric acid, butanol, acetone, isopropanol, carbon dioxide. The scaphophylline stain, that is 0.5% malachite green in water, can be used to distinguish endospores of Bacillus and Clostridium from other microorganisms. So this 0.5% malachite green uh, in water is used for the ready identification of Bacillus and Clostridium from difference in these endospores. There are commercially available polymerase chain reaction test kits, Bactotype, for the detection of C. perfringens and other pathogenic bacteria. So, coming on to the history. In the late 1700s, Germany experienced a number of outbreaks of illnesses that seemed to be connected to eating certain sausages. In 1817, the German neurologist Just, uh, Justinus Kerner detected rod-shaped cells in his investigation into this so-called sausage poisoning. So Germany was suffering from such kind of disease, and people were not able to understand what is causing this particular type of disease. So until 1870, the German neurologist Justinus Kerner, he detected that this particular type of disease is being caused by the uh, rod-shaped cells that is responsible for such type of poisoning. In 1897, the Belgian biology professor Emily van Emer, Ermengen published his findings of an endospore forming organism he isolated from spoiled ham. Biologists classified Van Amergam's discovery along with other known gram-positive spore formers in the genus Bacillus. So during that time, in the year 1897, the science and technology and even biology were not that much advanced. So people were making decisions based on their own theories, based on their own assumptions, and based on their own findings. Right? The technology and techniques were not that much advanced, and these people they had the understanding based on the techniques and technology available during the year of 1897. This classification presented problems. However, because the isolate grew only in anaerobic conditions, but bacillus grew well in oxygen. So they already faced a dilemma when they discovered that Clostridium are obligate anaerobes and bacillus are obligate aerobes, and they require a lot of oxygen for the growth. Circa in 1880, in the course of studying fermentation to butyric acid synthesis, a scientist surnamed uh, Prasmos- Prasmovsky first assigned a binomial name to Clostridium butyricum. The mechanism of anaerobe respiration was still not yet well elucidated at that time. So, people were not able to understand the complete nature of Clostridium, and Clostridium was extremely mysterious organism when it comes to bacteria, and they were trying to constantly study it. So, taxonomy of anaerobes was still nascent because the nascent it was in very starting phase. So the scientists were trying to explain it, but they were not being successful, and the study on anaerobes were in its starting phase. In 1924, Ida A. Bankston separated Van amalgams microorganisms from the Bacillus group and assigned them to the genus Clostridium. By Bankston's classification scheme, Clostridium contained all the anaerobic endospore-forming rod-shaped bacteria except the genus Sulfotomalacum. So, Clostridium difficile. So, diarrhea colitis are formed by the are as a result of this Clostridium. Pseudomembranous colitis exude on colon walls. So, it produces toxin A commonly, enterotoxin that attracts neutrophils and increases intestine wall permeability. So, it creates hole in the intestine, epithelial tight junctions in colon. So, so what it does, the toxin B destroys the enterocyte cytoskeletal integrity. So, it readily disrupts the cell cytoskeletal integrity at cellular level. Binary toxin CDT, adherence and cell death, eosinophil suppression by some strain. So, what they do, what is their specialty? They cause diarrhea and colitis, but what is their specialty? Their specialty is that they produce uh, toxin A, toxin B, and binary toxin. So, what is their basic nature? Their basic nature is to disrupt and disturb the essential formation, the essential construction of the intestine. They readily damage the cells or the epithelial tight junctions that are present in the intestinal walls. Okay, and the cytoskeletal integrity is affected readily, right? So because of it, there is a lot of change, and because of it there are many things that make them extremely dangerous in nature. Clostridium tetanus, spastic paralysis, lockjaw, grimace, uh, opithos or tonos, fever, sweating, back extension, cephalic tetanus, central nervous Placey, maternal tetanus associated with pregnancy, neonatal tetanus, infection spreads from the umbilical stump. So, tetanospasmin, teat liable neurotoxin, blocks the release of inhibitory neurotransmitter. A wound debridement, metronidazole, tetanus, antitoxin, and vaccinations are the solution of it. So it causes a very grim condition in the children, and Clostridium tetani causes tetanus. So these are all the working mechanism of tetanus. Clostridium perfringens. It is a gram-positive spore-forming obligate anaerobe. The basic mechanism is that it causes food poisoning. Food poisoning, typically mild symptoms, last for about a day, but it resolves without antibiotics. Okay, because most of the time the food which is already containing the spores of Clostridium uh, perfringens, uh, it gets incubated and turns into bacteria. But when it is eaten, it readily releases its toxins in the intestine, and that condition readily transforms into a food poisoning scenario. Clostridial myelin process. It is also known as gas gangrene. Can be deadly within hours without surgical excision. Penicillin 5 and hyperbaric therapy, 100% oxygen. So, gas gangrene is another disease that affected many during the American Civil War. During that time, the penicillin or antibiotics were not there, and the concept of antibiotics and sterilization had not yet emerged in the surgery or microbiology. Clostridium botulinum, descending flaccid paralysis, blurred vision with dilated pupils, dry mouth, constipation, respiratory failure. Okay, so these are the very dangerous symptoms or dangerous conditions that are a result of botulism. Botulism toxin A to G. A, B, E, most common toxins are performed, especially in canned foods, alkaline and aerobic conditions. Neurotoxin is absorbed from the gut and enters motor neurons. Block uh, ACH releases at peripheral motor junctions. So, metronidazole or penicillin or botulinum antitoxin or gastric irrigation. Boil home canned food. Discard damaged cans. Honey. Do not give honey to the infants that are less than 1 year old. So, mechanism of action. The result is unopposed uh, excitation of spinal neurons and muscles contraction leads to violent spastic paralysis. So it blocks the neurotransmission in your central nervous system. Tetanus toxin can also block uh, sympathetic preganglionic neurons, resulting in autonomic dysfunction. Finally, tetanus toxin may bind directly at neuromuscular junctions and cause neuromuscular facilitation. So you can see that it blocks the signals at the spinal cord periphery, and it is a very indifferent conditions when the patient is suffering with tetanus. Treatment in general, the treatment of clostridial infection is high dose of penicillin G, with the organism has remained susceptible. Clostridium welchi and Clostridium tetani respond to sulfonamides. Clostridia are also susceptible to tetracycline, uh, carbapamins, uh, metronidazole, vancomycin, and chloroamphetamine. The vegetative cells of Clostridia are heat liable and are likely killed by short heating at temperature 72 to 75 degrees centigrade. The thermal destruction of Clostridium spores requires high temperature above 121.1 degrees centigrade, for example, in an autoclave, and longer cooking times than 20 minutes, with few exceptional cases of less than 15 minutes recorded in the literature. So, Clostridia and bacilli are quite radiation resistant, requiring dosage of about 30 kgY, which is a serious obstacle in the development of shelf-stable, irradiated foods for general use in the market. The addition of lysozyme nitrate, nitrite, and propanoic acid salts inhibit Clostridia in various foods. Fructooligosaccharides, fructans, such as inulin, occurring in relatively large amounts in a number of foods, such as chicory, garlic, onion, leek, uh, artichoke, and asparagus, have a prebiotic effect, selectively promoting the growth and metabolism of beneficial bacteria in the colon. Such uh, bio, bifidobacteria and lactobacilli, while inhibiting harmful ones such as Clostridia and Fusobacteria and bacteroids. General properties of Clostridia. General properties of Clostridia include Clostridia uh, show following morphological properties. Most species are anaerobic, but few species such as Clostridium tertinum and Clostridium histolyticum, etc., are aerotolerant. Adherents can grow on agar even in presence of air. Clostridia are rod-shaped bacilli, meaning 3 to 8 and 0.4 to 1.2 m in size. The bacilli are highly pleomorphic and such long filaments and involution forms. They are gram-positive, but old cultures may appear gram-variable and even gram-negative in nature. Most Clostridium species, with few exceptions, for example, Clostridium perfringens, Clostridium tetani type 6, are motile due to presence of uh, Petricus flagella. Most Clostridia except Clostridium perfringens and Clostridium lutricum are non-capsulated. Ability to produce endospores in the is the most important feature of Clostridia. Spores. Formation of spores is variable depending on the species. Clostridium sporogens and other Clostridia sporulate readily, while Clostridium perfringens while and other species produce spores inconsistently. Sporulation takes place outside in the environment and also in animal bodies, but not in humans. The shape and location of spores in the bacteria vary in different Clostridial species, like central spores in Clostridium bifermentans, giving the bacillus as spindle shape. Subterminal spores in Clostridium perfringens, giving the bacillus aculeus shape. Over-terminal spores in Clostridium terminus, giving the bacillus tenis racket shape. Spherical terminal spores in Clostridium tetani, giving the bacillus drumstick presence. Here you can see the differences in the spores. Spherical terminal spores, Clostridium tetani, oval and terminal spores in Clostridium tertium, and oval and subterminal spores in Clostridium perfringens, and oval and central spores in Clostridium biformentum. So this shows the arrangement of the different kind in different variety of Clostridium. Spores are relatively more resistant forms than vegetative forms of the bacilli. They show a variable degree of resistance to heat, drying, and disinfectants. Spores are killed by 1% aqueous solution of iodine and 2% uh, glutaraldehyde at pH 7.5 and 8.5. They are particularly resistant to phenolic disinfectants. The spores survive in 2% formaldehyde solution even for up to 5 days. Clostridium botulinum spores can survive boiling at 105 degrees centigrade for 3 to 4 hours, while spores of Clostridium perfringens and Clostridium tetani are rapidly destroyed by boiling for less than 5 minutes. Some strains of Clostridium tetani can resist boiling for 15 to 90 minutes, and Clostridium perfringens type A strains, however, survive boiling for several hours. Culture. Most species are anaerobic, but few are, such as Clostridium triticum and Clostridium histolyticum, etc., are aerotolerant and hence can grow on agar in presence of air. Presence of adequate quantity of low redox potential substances in the media is important for the growth of, of anaerobic bacteria. Use of reducing substances such as ascorbic acid, uh, glutathione, unsaturated fatty acid, cysteine, thioglycolic acid, alkaline glucose, or metallic iron maintain low redox potentials in the medium. Most Clostridia grow at an optimum temperature at 37 degrees centigrade and pH 7 to 7.4. Growth on solid media is variable. Some Clostridia species produce hemolysis on blood agar. Robertson cooked meat (RCM) broth is useful medium for the growth of Clostridia. The medium contains unsaturated fatty acid, digested meat, and uh, sulfhydryl compounds. Clostridia grow well in the medium, rendering the broth turbid. Saccharolytic Clostridia turns the meat pink, while proteolytic species turns the meat black and produce foul smell. Most Clostridia produce gas. Now coming on to the MCQ, which of the following types of Clostridium perfringens alpha toxin is mostly abundant? The correct answer is A, type A. Which of the following is predominantly proteolytic? Correct answer is C, uh, Clostridium sporongens and Clostridium histolyticum. Which of the following properties are characteristics of tetanospasmin? The correct answer is D, all of them. It is heat liable protein, it is a neurotoxin, it can be toxoided. Type 4 type of exotoxin most commonly associated with botulism in men is or are type A, type B, type E, or all of these. Naegeli's reaction is the use for the identification of Clostridium perfringens. Most toxic exotoxin is uh, botulinum toxin. Which of the following species of Clostridium predominantly saccharolytic? The correct answer is D. All of them. Stromy clot reaction is used in identification of uh, Clostridium perfringens. Food poisoning strains of Clostridium perfringens belong to uh, opsonate type A. Uh, Redding of meat in cooked meat broth is produced by option C, Clostridium perfringens. These are the references from where we have collected the material. Thank you very much for listening to my lecture.